distinguished lecture series. Uh, and I'm very pleased to welcome today's speaker, Professor Jack Sostak from Harvard. Uh, Jack really needs no introduction, so I will not spend time on that. But what I will say is a little bit about the distinguished lecture series. This is a lecture that we organize once a month. And this is a public lecture, and some people have been asking that. Yes, it is a public lecture. Uh, and The previous speakers have been Vishwa Dikshit from Genentech, uh, Jim Collins from MIT, Rafi Ahmad from Emory, and now we have Jack today. So please tune in for future lectures as well. And these lectures also go on our website on the Trivedi School of Biosciences website and the YouTube channel. So with that, Jack, I welcome you and I hand it over to Ashok. To introduce Ashok, Ashok Trivedi is the founder trustee of Ashoka University and the founder of the Trivedi School of Biosciences. Ashok, over to you.
Thank you. Thank you, Shahid. First, I want to thank Professor Jack Sostak for giving today's lecture in the Distinguished Lecture Series. I met Jack during summer of 2019 in his office at the Mass General Hospital in Boston. We spent about 2 hours mainly discussing the upcoming School of Biosciences at Ashoka University. And towards the end, Jack agreed to come on board as a scientific advisory board member of the school. Later, we had a dinner at a nice seafood restaurant in Boston. Professor Jim Collins from MIT joined us there. Jim is also a scientific advisory board member of the school, and some scientists consider him to be the father of synthetic biology. During dinner, our conversation gravitated to the key focus of the upcoming school. I vividly recall Jack saying that to make it a great school, which should strongly emphasize research. Jim, who has won many teaching awards, emphasized teaching excellence. So with two eminent bioscientists at the table, it was clear to me we should follow the advice from both. And that's what we are doing. Jack was advisor to many PhD students at Harvard. Most notable is Jennifer Doudna, who got the Nobel Prize last year for CRISPR editing technology. Earlier this year, Walter Isaacson, who has written books about great innovators from Leonardo da Vinci, Albert Einstein, Steve Jobs, published a new one. This book is called The Code Breaker: Jennifer Doudna, Gene Editing and the Future of Human Race. It's a page turner that I highly recommend. And the next great book, just might be about the work that Jack Sostak is doing with his own his Origin of Life research team, at Harvard. Some scientists, including one Nobel laureate, believes that for this work, Dr. Sostak may win a second Nobel Prize, which would make him the Magnificent Four double Nobel winners into the Magnificent Five. Maybe that's why today's lecture has been the most popular one so far, with over 1,000 people registered. Now, Jack will tell us about his latest findings very, very shortly, and about his topic, why did life start with RNA, not something else? Very intriguing topic. Thank you for being here, everyone. And over to you, Shahid.
Thank you very much, Ashok. And with that, I just invite Jack to please tell us about why RNA.
Okay, thank you very much, Shahid and Ashok. It's a pleasure to be here and talk to everybody this morning or this evening, depending on where you are. So yeah, what I want to talk about is why RNA is so important in life, and why a little bit about why we think life began with RNA as the first genetic polymer. So the image that you're seeing on the screen now is just a schematic illustration of what we think a very early or primitive cell might have looked like, basically a membrane compartment, and you can see around the edge with the small bits of genetic material on the inside, which we think were RNA or something very close, at least, to modern RNA. So I will talk a little bit about that, but what I really want to focus on is, why is it RNA and not something else? And as you'll see, there are in principle many possible alternatives, but somehow RNA won out in some kind of primordial competition. So before we get to that, though, let me talk a little bit about RNA. And it's hard these days not to begin talking about RNA without at least mentioning the COVID-19 virus with its 30 kilobase RNA genome. And it's just really kind of stunning that a relatively small bit of RNA, 30 kilobases of genetic information coded in the sequence of an RNA molecule, could pretty much bring the world to a standstill and really cause unimaginable suffering and probably at this point millions of deaths. On the other hand, what we have also seen is that we can fight back against viruses like this with a new category of vaccines that are also based on RNA, on messenger RNA, that can be made enzymatically and synthetically on an industrial scale. This is a messenger RNA that codes for the spike protein of the virus. It can be encapsulated within a very special and sophisticated lipid nanoparticle, which efficiently enters cells, releases the RNA, which then causes the synthesis of spike protein, which then will lead to the production of antibodies and immunity to the virus. So I think these two things have really made RNA, which used to be kind of the area of specialized scientists studying the molecular biology of life, it's really made RNA into something that is everybody knows about now. It's become public knowledge that RNA molecules are incredibly important and can hugely affect how we live. Now, of course, RNA molecules do a lot more than what I just said. And over the last decade or two, it's become apparent that RNA molecules play a much broader and more diverse role in terms of different functions in biological cells than we used to think. So of course, it was known for many decades now that RNA molecules are important for protein synthesis. So we have the tRNAs that bring amino acids to the ribosome. We have the ribosome, which has several large RNA components. And then what, 30 odd years ago, it was discovered that actually RNA molecules can act like enzymes. They can catalyze reactions. Subsequently, it was found that there are RNA molecules called riboswitches, which are regulatory molecules. They can bind and sense and change in response to small molecules or proteins. And there are long non-coding RNAs that carry out very important regulatory. Okay, somebody is drawing on the slides, and I'd appreciate it if people didn't do that or could remove those marks. What was I saying? Yeah, so RNA plays an incredibly wide and important variety of roles in modern cells. The most important, arguably, is the role of RNA in the ribosome. Okay, it would be really nice if we could get rid of these red lines on the slides. I can't do it from here. Here is a diagram of a ribosome, the large subunit of ribosome. This is from a crystal structure that was determined by in the Stites lab quite a long time ago now. And the gray squiggles that you see are RNA molecules, and these golden squiggles are proteins, ribosomal proteins. And what you can see, this little green area in the middle is an active site inhibitor. So this is the location within the huge ribosome where proteins are actually synthesized, where the peptide bond is made. And you can see it's surrounded just by RNA. So it turns out, based on this structure and lots of other work, that it's actually RNA molecules that synthesize all of the proteins in our bodies and in all cells and viruses in all of biology. And so the logical conclusion from that is that, thinking about the evolution of life, RNA must have come first, right? Because RNA is the thing that started to make proteins, which subsequently became so important in catalysis. Okay, let's see. So let's have a closer look at RNA. Yeah, can I just say it would also be helpful if everybody else could mute themselves so that there'll be less background noise, please. Okay, let's have a closer look at RNA, see what it actually is at a chemical level. And so what you can see here, so don't worry about the details, but what we can see, the blue parts are the nucleobases, the A, C, U, and G. And these are the molecular recognition units, and information is coded in the sequence of these A, C, U, and G building blocks. Every one of these is hooked up to a sugar shown in green, and in RNA, the sugar is ribose. And the ribose units are linked together by phosphates to make this polymer, which can be many, many thousands of units long. But it's basically a repeat of this simple structure that you see here. So there's a puzzling aspect to this, though, which is, this is a pretty complicated molecule in terms of its chemistry. And for a long, long time, it was almost impossible to imagine how something like this could be made in a non-biological way on the early Earth in order to be the genetic information of the first cells. So it was a huge puzzle as to how RNA could have originated. And there's been a huge amount of progress on that question over the last 2 decades. And I'll talk very briefly about some of that, but I don't want to go into too much detail of the complicated chemistry, but just to say that you can break down a really complex structure into simpler and simpler components. And it's been possible to work out very robust pathways that build up the complexity of the building blocks of RNA and the structure as a whole. It's not an entirely solved problem. I just want to say there's been a lot of progress. But along with that progress comes the recognition that there are a lot of possible variations on this structure. And so RNA is not the only thing that, that might be made on the early Earth. And the first person to really systematically look into that question was the great chemist Albert Eschenmoser, who, starting back in the 1980s, actually, really tackled this question head on by chemically synthesizing a whole range of nucleic acids that are similar to RNA, but have different structures. And this diagram is taken from a review article that Albert wrote about 10 years ago, and illustrating, in this case, focusing really on variations on the sugar parts of these molecules. So here in the middle, we have the building block of RNA, and the ribose sugar is filled in with red. But there are many, many, many possible variations on the structure of the sugar, and also on the way that the connections between adjacent units in the chain can be made. And Albert and many people in his lab synthesized over a period of many years all of the variants that you can see here. And it turns out that the ones that are colored in actually can, in principle, act as genetic polymers. They have Watson-Crick base pairing. So A pairs with U, G pairs with C. They make a double-stranded duplex where the strands are anti-parallel. That is, they go in opposite directions, just like in DNA. And the interesting thing is, so if you look at the ones that are in red, these are polymers where a sequence of one type can base pair with the sequence of the other type. So for example, we've known for a long time that DNA sequences can pair with RNA sequences. And there are two other molecules. One over here is Threo's nucleic acid that I'll talk about quite a bit later on. And all these red ones can base pair with each other. All of these blue ones can also base pair with each other, but not with the red ones. And this one over here, HomoDNA, can base pair with itself, but not with any of the red or blue structures. So not only are there a huge number of potential alternatives to RNA that are still, double-stranded Watson-Crick type duplexes, and so they could act as genetic carriers of genetic information. But there are different classes that could be independent of each other. And so this huge amount of chemical synthesis and structural characterization really lets us focus in on the question of, well, why, in fact, is biology based on RNA and not on, for example, Threo's nucleic acid or this Lixo's peranecyl nucleic acid or any of these others? So that's the central question that I want to talk about. Why is it RNA that won out, and why isn't biology based on one of these other structures? Okay. So in the interests of time, and also because some of these alternatives are more interesting and plausible than others, I want to focus on two potential alternatives to RNA. And these can be abbreviated as ANA and TNA. So as I said before, in RNA, the sugar here is ribose. And by analogy, in ANA, the sugar here is a sugar called arabinose. And you can see the only difference between ribose and arabinose is that this hydroxyl is down in ribose and up in arabinose. So it's a stereoisomer. It's a very closely related structure. And we can ask, and well, we actually know, I'll show you, that if there's a chemical process that would lead to the synthesis of RNA building blocks, it would almost certainly also lead to the synthesis of some of these arabinose-based building blocks. Over here is TNA, which stands for Threo's nucleic acid. And this is a little bit more different, although it might not be obvious until you look carefully. But this is a sugar which has 4 carbon atoms, and these two have 5 carbon atoms. So the chain is shorter. Every linkage is 5 atoms instead of 6. And yet it can still make the standard Watson-Crick base paired duplex structure. So just thinking about these 3 very similar nucleic acids, we can again ask, why is biology based on RNA and not on ANA or TNA? Okay. Another very closely related question is, well, if we have all of these alternative genetic materials, and they might all be made by very closely similar kinds of chemistry going on the early Earth before, the origin of life, what would the first genetic materials have looked like? In other words, could primitive RNA have been a kind of messy structure that wasn't as uniform and homogeneous as modern RNA? So RNA made in a biological system, it's made by enzymes. Everything is very closely controlled. All the linkages are the same. The nucleobases are the standard nucleobases, A, C, U, and G. But if we think about, uncontrolled chemistry going on the early Earth, we're pretty sure that you would have all kinds of possible variations. So for example, the connectivity between units might be from the 3' hydroxyl to the 5', as we see in biology. Or it might be from the 2' hydroxyl to the 5'. And we know this can happen. And similarly, maybe mixed up with ribose-based building blocks, we'd have some of these arabinose-based building blocks or threose-based building blocks. Maybe the primitive nucleic acid was a very heterogeneous mixture of different kinds of building blocks. And in that case, how did modern RNA, which is very homogeneous, arise from this very messy primordial mixture? And of course, we can ask the same thing about the nucleobases. Maybe there were alternative nucleobases that had, for example, sulfur atoms here instead of oxygen. Maybe we have this structure, inosine, instead of guanosine, and so on. There are many possible variations. And we can also ask about different kinds of connectivity. So there's a lot of variation that could have existed early on. And if that's the case, we'd like to understand how modern homogeneous RNA emerged from this primordial mess. Okay, so in order to start looking at that, let's have a closer look at the standard building blocks of regular biological RNA. So these are cytidine, uridine, adenosine, and guanosine. So C, U, A, and G. Again, the sugar in these molecules is shown in green, and this is the ribose sugar hooked up to phosphate. So if these molecules could be made in spontaneous primordial chemistry, then they have to hook together to make an RNA chain. And to do that, this phosphate has to be activated. It needs what we call a good leaving group on the phosphate so that these building blocks can polymerize. Now in biology, that leaving group is pyrophosphate, so 2 more phosphates. So all of these are present in cells as nucleoside triphosphates, which enzymes can polymerize. Early on, before there were enzymes, we needed to have more reactive groups here so that the polymerization could happen spontaneously. Okay, so that's something that we've worked on, but I won't have time to go into that in more detail. But what I want to show you is just a little bit about how these molecules might have been made and why that makes us think that there are particular alternatives that might have been made at the same time. Okay, so this is another way of thinking about this idea that prebiotically, before there were enzymes, things would have been very messy and heterogeneous. And so the late Leslie Orgel and his colleague Jerry Joyce, many years ago now, kind of made fun of the idea that on the early Earth, you could have some nice warm prebiotic pool filled with pure building blocks of RNA. I mean, it's just chemically not realistic. So they're saying, well, look, we have to either find a simpler predecessor of RNA, something easier to make, perhaps, or we have to figure out how modern homogeneous RNA could have emerged from some kind of early mixtures. Okay, so this is the problem again, just to restate and show it as a diagram. So the idea is that we would have multiple different types of building blocks synthesized by spontaneous chemistry, prebiotic chemistry on the early Earth. And we would not only make the building blocks of RNA, the ribonucleotides, but we would also make at least some of these other types of building blocks, arabinonucleotides, threonucleotides, even deoxynucleotides, the building blocks of DNA. And if these are all made and mixed together and started to assemble into short chains, they'd be very messy and heterogeneous. And from such heterogeneous structures, it's very hard to imagine how anything useful would emerge. So that's the puzzle. Okay, so how do we start to get at that? And then a really important thing to understand is some of the basic aspects of how these building blocks of RNA might have been made. So if you look at one of them, so this is C, which we showed you before. So it has the cytosine nucleobase, the ribose sugar, and a phosphate. And you try to think, well, how would you make a molecule like this? And the kind of obvious thing is, well, maybe you'd make the nucleobase first, you'd make the sugar separately, and you'd have some phosphate around, and then somehow you'd put all these things together. And that's what people thought for a long, long time. And the problem is it just basically doesn't work. So what's the alternative? Okay, so if you try to hook up ribose and cytidine to make cytosine, it just doesn't work. So how can you get around that problem? And so this chemistry actually goes back quite a long time to early work from, again, Leslie Orgel, but Matt Powner and John Sutherland in a breakthrough paper in Nature over 10 years ago really worked out a very efficient way that you can get around that problem, which is, so here's the bond that connects the nucleobase and the sugar, right? And this is the one that's very hard to make. But it turns out if you make that bond first in this odd looking little compound down here, which is very easy to make, you can make this and then build out the rest of the nucleobase and the rest of the sugar, and you can get to the desired building block of RNA. Okay. the trouble is that chemistry also gives you some closely related molecules. So this is the one that we want to make. So where the nucleobase is above the ring of the sugar, and the sugar is ribose, but that same chemistry will give you some of the arabino building block, and it will give you related molecules where the nucleobase, instead of being on top of the sugar, is below the sugar. And so again, we have this question: if all of these different kinds of molecules are being made, how do we end up with just the one that we want, or at least with polymers that are RNA made of the building blocks of RNA? Okay. So let me try to explain very briefly why we think these particular alternative building blocks would have been made. So if we think of ribose, shown again in green here, is made it's a sugar that has 5 carbon atoms. And the way that sugar could be put together is by starting with a much simpler 2-carbon sugar, shown here, reacting with this compound to make this intermediate, and then in a subsequent step, bringing in a 3-carbon sugar. This is glyceraldehyde. And so a 2-carbon sugar plus a 3-carbon sugar give you the 5-carbon sugar ribose, but it can also give you, depending on how they come together, the 5-carbon sugar arabinose. Now this is an intermediate, and so 2 more reactions give you the actual nucleotide building blocks of RNA and ANA. But from this chemistry, you can see that these stereoisomers, these very closely related compounds, are likely to have been made at the same time. Okay. Now, there's a difficulty with this pathway, which is that it's a little tricky to figure out how to first bring in the 2-carbon sugar and then separately, in a subsequent step, bring in the 3-carbon sugar to build up the molecules you want. And wouldn't it be much, okay, so sorry, this is just showing the end products again, ANA and RNA building blocks, differing only in the position of this hydroxyl. So wouldn't it be much easier to make a building block where you only need 1 starting sugar, the 2-carbon sugar, shown here, glycolaldehyde. And so we could make this intermediate, starting from 1 molecule of this 2-carbon sugar, and then bring in a second molecule of the same sugar and make this Threo's intermediate, which would go on to make the building blocks of TNA. This seems much easier, right? And that really, I think, brings home and emphasizes the question, well, if threonucleotides and TNA are easier to make than RNA, why do we end up with a biology that's based on RNA and not TNA? It's really quite an interesting puzzle. Okay, so again, here's the comparison. Here's our standard ribonucleotide. That's the building block of RNA. And here's a threonucleotide, the building block of TNA. And the only difference is that this carbon here is missing in the Threo's building block. Okay, so we have these 3 very similar nucleotides that could be made by very similar chemistry. Why is it that RNA won out in the end? And the approach that we've taken to gaining some insight into that is to think about the chemistry that's involved in actually copying sequences. So if a primitive cell has a sequence that does something useful, that sequence has to be replicated, right? And exactly how that works, we still don't fully understand. But at some level, there has to be chemistry that can copy a template sequence. So if a template sequence is a sequence we want to replicate, the first step in replicating it is just to copy it. And a really nice model system for studying that is to have a little piece of RNA that will base pair with the template. So we call this the primer. We bring in these these monomers, these nucleotides I've been showing you, and they get added to the primer spontaneously. And the primer grows and makes a copy of the template strand. Now, it's probably more realistic if we're thinking about prebiotic mechanisms to think of monomers and short oligomers binding to a template and all gradually getting hooked up together. But the chemistry would be more or less the same. Now, it does turn out that the chemistry by which a primer grows and copies a template strand is kind of surprising and not what we thought for many decades. And so here's shown again in a simplified schematic diagram what we think goes on in copying. So we have a short piece of RNA that's a primer bound to a longer piece, the template, and we want to copy this region here. And activated nucleotides come in, they bind by Watson-Crick base pairing to the template, and the primer grows by reacting with these monomers. And we used to think, everybody used to think that monomers would come in one at a time and the primer would grow by reacting with monomers. Well, it turns out that's actually not how it happens. And we worked out, or I should say Travis Walton, a graduate student in my lab, worked out a few years ago that what actually happens, at least most of the time, is that these types of monomer building blocks react with each other to make this more complicated structure, which has two nucleotides in it. And this dinucleotide binds to a template. And this bridge structure here is very structurally organized. It's oriented perfectly to help make this reaction go. So the primer reacts with the adjacent nucleotide, grows by one nucleotide, and then you kick out another nucleotide as a leaving group. So in order to copy a template, you need several different things. You need a primer that can grow. You need these activated building blocks. You need them to form this dinucleotide, and you need a template. And we can ask, in each of these situations, what happens if we replace the RNA or the ribonucleotide with one of these alternatives? Okay. Now, the chemistry that I've just shown you is worked out and confirmed in many different ways. But one of the most beautiful and to me the most satisfying ways of looking at it is by X-ray crystallography. So we can actually get snapshots of what these molecules look like in 3 dimensions. And we can actually see in a series of crystal structures that were determined by Wen Zhang when he was a postdoc in the lab, we can see these sequential reactions happening. So in this image, we're only showing you a part of an RNA primer template complex. We've soaked in activated monomers, which are here. Here are 2 monomers that are actually base paired with bases on the template. Here's the leaving group, which we don't see in the crystal structure because they're in many different positions. But after about an hour, we see that these 2 adjacent nucleotides have reacted with each other, and this bridge structure has formed. And then if we wait longer, we can see that this bridge structure has broken, and now the primer has reacted with the adjacent nucleotide, and the primer has grown by 1 base. So we can see this series of chemical reactions happening in a series of crystal structures. Okay, so with all of that as a kind of background for how copying chemistry actually works, let's have a look at what happens when we try to make this work with some of these alternatives like ANA or TNA. And so, as I mentioned, we can take the RNA structure and replace individual nucleotides with, in this case, arabinose nucleotides or Threo nucleotides or almost anything else that we can actually chemically synthesize in the laboratory. And we can ask then, how does the copying chemistry work? How is it affected? Is it inhibited? Does it work fine? And so on. So what we've done to look at the possibility of oops, sorry, let me go back. What we've done to look at the effect of replacing little bits of RNA with arabinose nucleotides is to replace the nucleotide at the end of the primer with an arabino nucleotide. And what we observe when we do that is that the primer extension is like 100 times slower. So everything else can be RNA, but if you just replace this one nucleotide at the end of the primer, it doesn't like to react with the monomers or this dinucleotide intermediate. On the other hand, if we replace the incoming monomers with arabino nucleotides, we form this reactive intermediate, this dinucleotide, just as well. That step is not really affected. But the reaction, once this intermediate forms and binds to the template, the reaction with an all RNA primer is way slower again, 10 to 60 times slower. Okay, so replacing parts of the system with ANA really, really messes up the copying chemistry. And the interesting and somewhat surprising thing is that you can replace bits of the template with ANA, and the effect is quite modest. So it can be only maybe a factor of 2 slower or 5 or 6-fold slower, depending on the change. So what all this means then is that if you had a heterogeneous template that had some ANA nucleotides, it could be copied over and make an RNA copy just fine. But if you start to put ANA building blocks in and trying to do the copying chemistry with them, it doesn't work. And in fact, we've done competition experiments where we have both, for example, ribonucleotides and arabinonucleotides. And we let the reaction go, the copying chemistry go, and then ask, well, what actually got made? And what we see is that what's made is essentially all RNA. We can't even detect the small bit of ANA that might be made. Okay, so clearly in this competition, RNA wins. And so then we can also ask, like, why is that? You know, chemically and structurally, what's actually going on here? Why is it that RNA is better? And so again, to get some insight into that, we grow crystals and we solve the crystal structure and try to get some insight as to what's different about the structure that might explain this. And in this case of putting an arabinonucleotide at the end of the primer, what we see is that the hydroxyl that has to react with the phosphate next door it's further away than it would be if this was a ribonucleotide. And there are some changes in the geometry of the sugar that also make it look like it would be less reactive. So we can get some insight into why RNA is actually better in a chemical sense. Okay, what about the other alternative that I mentioned, TNA, which is made from Threo's nucleotides? So this is the one that actually looks much easier to generate, and yet it seems to be something that could act as a genetic polymer. So why isn't biology based on TNA? Why did RNA win out in this case? So we do the same kinds of experiments, right? We make our RNA primer with a TNA base at the end. We can make the template with TNA as in the place that can be copied over. We can put in the activated Threo nucleotide building blocks and watch the copying chemistry go. And what we see here is that when there's a TNA nucleotide at the end of the primer, it reacts more slowly, but it's actually only 5 to 10 times more slow. It's not as bad as the ANA situation. Interestingly, things are different when you look at the Threo nucleotide building blocks. They do react to form this dimer, but that reaction is slower. And once that dinucleotide building block is made with TNA units, it reacts almost 100 times slower with an RNA primer. It can be more reactive if one side of these is RNA, but if they're both TNA, it's very bad. So once again, in the copying chemistry, if you bring in TNA nucleotides, it's much slower, and RNA would outcompete TNA by a lot. And interestingly, again, we see that if you put TNA building blocks in the template, they can be copied over, and there's only a small effect in the rate of copying. So if you have a heterogeneous template, you can copy it and make RNA. But if you try to mix in TNA nucleotides, they're much less reactive. And so again, RNA would win in a competitive situation. And just like before, we can grow crystals, solve the structures, and try to get some insight into why RNA is better at doing this copying chemistry. And so here's a little fragment of the reactive complex where everything is RNA. And here's the 3' hydroxyl at the end of the primer that has to attack this phosphate to make a new bond, so the chain will grow. And we have a certain distance here. But if you make this intermediate out of TNA building blocks, this distance now is much greater. And that really, I think, explains why this reaction with TNA is so much slower. And it's actually a very simple thing to understand. So in RNA, the reactive phosphate is held away from the sugar because there's a carbon atom in between. In Threos, the phosphate is kind of pulled back and it's right over the sugar. So it's been pulled away from the end of the primer, which it has to react with. So I think from this kind of structural information, we get a very clear picture of why RNA is better at doing copying chemistry than TNA is. Now you might think, well, what if you made everything out of TNA? Maybe then it would work. But we've done that, and it's actually even worse. So TNA really is extremely bad at doing copying chemistry, whereas RNA is quite good. Okay, so we can also understand, again, from crystal structures, why heterogeneity in the template is not so bad. And the reason is that the nucleic acid backbone is so flexible. It has so many bonds that can rotate and shift around that you can basically have a structural perturbation, like replacing a bit of RNA with a bit of TNA, and it doesn't make any difference in the overall structure. It can be accommodated by the flexibility of the backbone. So the perturbation is very local, and I think that can explain in large part why you can copy over a TNA nucleotide almost as well as a ribonucleotide. Okay. So at the very beginning, I mentioned that there also can be variations in the backbone structure, such as having 2' 5' linkages instead of 3' 5' linkages. And what you see here again is a crystal structure of a duplex where there's a particular 2' 5' linkage. And it really looks just almost like a regular RNA duplex. So again, this change in the connectivity of the strand has only a very small local effect, which explains why it can be copied over. Similarly, you can have 2 phosphates instead of 1 in the backbone. Again, the perturbation is very local, and we've shown in experiments in the lab done by Tom Wright, largely, that you can copy over this kind of variation, this kind of heterogeneity. You can even have an amino acid bridging two nucleotides, and you can copy over that heterogeneity. So if you put all of this together, you come up with our current model, the way we're thinking about things, which is, as I said much earlier on, the prebiotic chemistry gives us probably mostly ribonucleotides, the building blocks of RNA, but it's also going to give some related molecules like arabinonucleotides, threonucleotides, deoxynucleotides. So we'll have a mixture of different kinds of building blocks, and these will assemble to make short strands of a very kind of heterogeneous, messy version of RNA. And what we think happens then is that since these are basically random sequences, every now and then, some of these short oligonucleotides will be able to base pair with each other just by chance, having a little region of complementarity. And then this is the setup that you need for primer extension to happen. So these chains can now grow. But the added nucleotides, as you've seen from the experiments that we've done, will be almost exclusively ribonucleotides. And so by going through this kind of cycle of primer extension and then having things come apart and then reassemble and doing a little more primer extension, you would gradually build up oligos that are almost exactly what we see in modern RNA, relatively homogeneous and made just by the chain of ribonucleotides, with all of these alternatives having been hugely outcompeted by RNA. So that's our current model. We still have a lot more work to do to test these ideas, but we think that this, together with a lot of other sort of steps that filter out alternatives, can provide an explanation of why biology began with RNA and not with any of these alternatives. And moreover, how homogeneous RNA could have emerged from these very messy, heterogeneous intermediate states. And I just want to end with a sort of philosophical question about this. And I don't usually like to engage in philosophical speculation, but I find it very interesting that out of all of the alternatives that have been looked at, and that's many more than what you see just here, it seems that RNA is by far and away the best. And the question is, does that mean that life would always emerge with RNA as the initial genetic material? Or is it actually possible that if life began independently somewhere else, so for example, on one of these exoplanets orbiting some distant star, maybe if the conditions were a little different, would one of these or some other alternative that we haven't thought of emerge as the basis of life? It seems odd to me that the chemistry seems so focused towards RNA as the kind of ultimate winner, both in terms of ease of synthesis and in terms of outcompeting alternatives. And so I think this is a huge question for the future, to really try to understand if RNA is the only way that you can get to life, or if there actually are alternatives that might be just as good, and we just haven't thought of them yet. Okay, so with that, I'd like to emphasize that all of this work and a lot of other work I didn't have time to talk about, it's been done by fantastic people in the lab. And so here I'm just showing some of the key graduate students who've done a lot of this work. So a lot of the early work on the mechanism of copying was done by Travis Walton. A lot of the work on comparing primer extension with different types of molecules was done by Chris Kim. And almost all of the crystallography that I showed you was done by Wen Zhang, a former postdoc in the lab. And so with that, I'd like to conclude. Thank you for your attention, and I'm happy to take any questions at this point.
Thank you. Thank you very much, Jack. I'm Shashi here. I'm supposed to moderate the question answer session today.
Okay.
And good to see you after a long time. So, so what I'll do is, so the participants, you please raise your hands, those who want to ask questions. And then, you know, I will have to unmute you. Before that, there are a couple of questions in the chat box. Maybe, you know, Professor K. N. Ganesh has asked a question. Can selects technique be used to select enzymes or polymerases that bind ANA or TNA in a selective way?
Yeah, that's a very interesting question. So actually, I forgot to mention that our work on TNA was done in collaboration with the laboratory of John Chaput. And John has been studying exactly that with TNA for many years now. And he has been able to find enzymes that can make and copy TNA so that he can now do CELEX experiments. And he's been able to evolve TNA sequences that act as aptamers and as catalysts. So those kinds of experiments suggest that, you know, TNA is at least almost as functional as RNA if you have the right catalysts to make and copy it. We haven't done things like that yet with ANA. Phil Holliger's lab in the UK has explored CELEX experiments with ANA and several other variants that are collectively known as XNAs. And in many cases, yes, you can use CELEX to evolve functional sequences.
So there are many questions related to ANA or TNA being in the living cell, or they can be translated. I think you partially already answered that question. Rohini, Chinta, and a couple others have mentioned that can this be found in living cells? Can they be translated into some protein information? You know.
Yeah, so basically they're not found in living cells. As far as I know, there are no examples of ANA or TNA. And the question of whether they could be translated by the ribosome is interesting. I doubt it, but there may be some. In fact, there are some other variations that we're actually looking at. We're interested to see if they could be translated, but we don't know yet.
Okay, maybe Samay Pandey, you want to unmute yourself? You want to ask a question yourself?
Yes. Thanks, Shashi.
Go ahead.
Jack, I realized that kind of the take-home message, and you might correct me if I'm wrong here, was that the reaction rate of formation of ANA or TNA or RNA was important. I missed out on the importance of fidelity. So how reliably you can regenerate the information. So is there a difference between the three molecules that you talked about? That maybe RNA is more reliable, that it can generate the exact same copy of RNA again versus TNA or ANA?
No, that's a great question. We don't have much data on that yet, but that is absolutely something that we should try to do. We should look at. So far, I have to say we're focused on the question of fidelity in RNA itself, because so far the fidelity of copying in an all RNA system is not so great. And so one of the big questions that we're trying to deal with is how can we make the fidelity of RNA copying better? And so we've been kind of more focused on that than asking about fidelity in some of these other systems where just the copying chemistry is so much more slow. But yeah, that's a great point.
Okay, Vijay Subramaniam, you want to unmute yourself? and ask you a question.
Yeah. Okay. Hello. Can
Go
you
ahead.
hear me?
Yeah.
Yeah.
Yes.
Thank you. So great talk. I was just curious about secondary structures from these alternates, and if the idea is that these might be precursors of early life at some point and got washed out, they should also have secondary structures to catalyze enzymatic reactions. That would be the purpose, right? Like if it is basis of life.
Right. So we know that TNA can do that with no problem. So you can make secondary structures because TNA can base pair with itself, and you can make complicated structures that fold up in 3 dimensions. And there are even TNA sequences now that will act as enzymes. Not very good, but they have some enzymatic activity. Now, ANA is interesting because ANA can pair with RNA, but it can't pair with itself, or at least not very well. So it's less clear if you could make secondary structures and ribozymes or aptamers with ANA. So I think that's something that needs a little bit more work. But yeah, that's, again, a great question.
Anchal Bharati, please unmute yourself and ask your question.
Hello.
Yes, go ahead.
Hi, sir. Yes, sir. So, sir, like we said, like RNA, we are relating RNA to the origin of life. So somehow we are actually saying that since for the origin, the organism, it need to progress, and when it will progress, it should have it as its genetic material. So if I say that RNA were a genetic material in organism and somehow reverse transcription took place and DNA took over it, then can I relate it to the first organism that was formed? Like, can we relate it to viruses which still have their RNA as their genetic material? Can we say that there were such conditions in the primordia that viruses were actually, they can be related to the origin of life and they were not obligatory parasites somehow?
Yeah, very interesting set of questions there. So I can only tell you the way we think of it, right? So we think that the very first cells are extremely simple and primitive, and would have had a genome composed of RNA, probably a very short genome. And in those primitive cells, there was not, the RNA could have been replicated first chemically, and then later on, presumably by ribozymes that made replication more efficient. And the question of when and why DNA arose as a more efficient way of storing larger amounts of information is still being debated. There's even the current idea that RNA and DNA might have arisen together as a kind of mixture and then bifurcated to make, on the one hand, pure RNA molecules that carried out functions and pure DNA molecules that stored information. So that's a very active area of research. Now, as for the origin of the first viruses, it's a little bit hard for me to imagine how a virus-like structure might have emerged before we had protein-based biology, because for membrane fusion events, these are not efficient unless you have good protein catalysts of membrane fusion. And it's a little hard to imagine how RNA itself could do that. For those reasons, I kind of think that viruses, and specifically RNA viruses, emerged at some later stage in evolution. On the other hand, I can say you could have RNA parasites that, one of the reasons you need to have a cellular structure, a compartmentalization, is to prevent parasitic RNAs from taking over and causing the whole system to crash. So if you have a membrane barrier, then parasitic RNAs can't get in there and for example, replicates so efficiently that the cell dies. So I think compartmentalization is important both for allowing evolution to occur and for preventing a crash due to parasitic sequences. And then only later, when you had proteins that could catalyze membrane fusion events, could viruses emerge. Anyway, that's how I think about it. Certainly not everybody agrees.
Okay, so Jack will take a couple of more questions. One is by Professor Ganesh, and later Ashok wants to ask a question. So Ganesh, I'll unmute you.
Yeah, it's a great question, Professor Zoltak. I just had one doubt about your first part of the talk. Second part of the talk. Your copying of alternatives, your TNA and ANA, for the copying mechanism, you were using enzymes which have got evolved in RNA world. Right?
No, sorry, there are no enzymes. All of this is just chemistry.
Oh, okay,
no
it's chemistry. Oh, that's fine. Somehow I thought that you had used enzymes, so that's what I was worried. Okay, thank you very much.
Okay.
Okay, Ashok. Ashok, you can unmute yourself. Unmute yourself, Ashok. No, it's yeah.
There
Go
we
ahead.
I'm still unmuted. Can I?
Yes.
Can you
Yes,
hear
I
me
can
now?
hear you now.
No,
Yeah.
Jack, I don't have any questions. Okay. I just want to thank you for such a wonderful lecture and taking the time to be here. And I think people have lots of questions, but I know your time limitations and so forth. If you can take one or two more questions, that'll be great. Otherwise, I really appreciate your coming and giving this lecture.
So shall I go ahead and ask one or two more questions, Jack?
Yeah, let's have two more questions.
Okay, great.
Sorry, I don't have to get going.
Ayush Mistry.
Hi, Professor. Am I audible?
Yes.
Perfect. Hello, Professor Rostak. Thank you for the talk. My name is Ayush, and I'm an undergrad at Ashoka. I just had one overarching question. As you initially mentioned, with the existence of an extremely messy pool of RNA and a lot of uncontrolled chemistry in the background that sort of went on in the primordial cell, and the fact that most of these alternative hypotheses on evolution rely heavily on a lot of molecular cooperation that is being continually revealed. As someone who intends to replicate these conditions in lab, how do you fit this high degree of chemical stochasticity in a controlled experimental setup?
Yeah, that's a great question. It's of course very difficult to, we don't know exactly what the primordial conditions were. We're actually trying to work out a set of conditions where we can actually see decent, rates and accuracy of copying, in the hope that will actually give us some insight into the geochemical environments that could have supported early life. So it's a kind of iterative process, of playing around with the conditions, trying to see what works, trying to see what's geochemically plausible, and then refining the picture in that way. And hopefully we'll converge on a picture that is both geologically and chemically plausible and actually works in terms of replication chemistry.
Great. Shantanu Chaudhary, you want to ask a question?
Yeah. Thank you very much, Professor Zostak. That was a beautiful talk, a great learning experience, I guess, for all of us. My question is regarding once the enzymes come into the picture, right? You have an enzyme-free situation where you see this. Once an enzyme comes into the picture and a catalysis comes into the picture, how do you see this next cycle of evolution going on, which will depend a lot on the selectivity of the enzyme for the particular ANAs or TNAs? Do you see things changing with a polymerase, for example, in the mixture of nucleotides?
Yeah. So again, I think the way we're thinking about it is that the intrinsic chemistry of these processes would lead to very primitive cells where replication that favors RNA is going on because of spontaneous chemistry. Now, that spontaneous chemistry is kind of slow and not very accurate. So I think the next step would be the evolution of a ribozyme, an RNA enzyme, that could gradually make the copying chemistry faster and more accurate. And that would allow more information to be stored and more different ribozymes that do useful things for the cell to emerge. And then much later on, when you have the evolution of translation, then you can have protein enzymes that are even better and can use more stable substrates like the NTPs and allow the evolution of these very large DNA genomes that we see in biology today. So I think it's a stepwise process like that. But to come back to your question, I think that once you have a cellular starting point that's based on RNA, that's kind of locked in to the system. And it would be very hard to switch to a different genetic material once you get started with one.
Narendra Mehta, you want to ask your question? Okay, if not,
okay.
We'll one last question from
last slide.
Yeah, Ujjwal Kumar Trivedi.
Hello, sir.
Yes, go ahead.
It is my pleasure to listen to Professor Jack, and I'm a big fan of you and your work on origin of life. And yeah, so most of the questions have been answered, and I'm really intrigued to know, has anyone tried the copying and extent polymerization mechanisms using L sugars?
Yes. So can we have a form of life that's basically a mirror image of the chemistry that we see now? And yes, in fact, Jerry Joyce's lab has, I think, done more work on that than anybody else. And as you'd expect, if you make everything out of the mirror image structure, so sort of mirror lRNA rather than dRNA, everything works just the same. And you only run into problems when you try to combine them. And so again, that comes back to one of the big mysteries in the origin of life, which is when was homochirality first established? I think it had to happen before the emergence of biology, and there are now multiple physicochemical mechanisms that in principle could lead to having an all L or all D system. But this exactly how and when this happened is still quite a mystery. So a lot of people are working on that. It's a very, very interesting question.
Thank you.
But basically, either mirror image form works just as well.
Thank you so much, sir.
Yeah, great. Thanks. So in the interest of time, Jack, maybe we'll stop now.
Yeah.
I know there are at least another 10 questions in the chat box, and at least 9 people have raised their hands. Apologies to all of them. But perhaps at some point they can watch many of your other lectures on YouTube and other sources. And definitely would like to bring you back for one more longish kind of a lecture, and hopefully on our campus.
Yeah,
you have been a great advisor, and you have helped us recruiting many faculty in Ashoka. And I hope someday we want to bring you to the campus and showcase what's being done in Ashoka and also elsewhere in the country, the chemical biology work, particularly on the chemical origin of life.
Yes. Well, thank you. Yeah, I would love to visit Ashoka.
Thank you very much for giving your time for us. And thanks to Ashok, he's always been a big supporter and a motivator for all of us. Thank you. Thank you very much.
Great. Thank you. Thank you.
And the audience, thank you very much. And please wait for the next distinguished lecture in this series, and hopefully we'll bring another great speaker and great topic.
Great. Thank you very much. Thank you everyone for listening, and thanks for the great questions. And,
stay safe.
Yeah. Stay safe. Thank you. Bye.